It's pretty hot here in Oakland. Um, so sorry if I'm just sweating the whole time here. Forgive me. But hey, everybody, it's me, Josiah Zahner, and we are on Bioengineering 102, Class 4. Today we're going to learn about last week, hopefully, what you did was you grouped some bacteria and you purified your plasmid and you ran out of gel to verify that your plasmid was actually there, right? So this week, what you're going to do is, now this is one of the most accurate forms of sequencing, um, and generally it's always used for short sequences. So when you're just trying to sequence, um, 800 or 1,000 base pairs or something, you can't sequence much more than that generally with Sanger sequencing. So this week, download SnapGene if you haven't already. There's a link in the Google Classroom, and there's a document submitting and analyzing DNA sequencing that you need to follow. So, first thing, when you run a gel, you know, feel free to use, um, like 5 or 10 microliters or something like that. But if it doesn't work, be wary that if you use all your plasmid DNA sample, you're not going to have any left for sequencing. And even if you do have some left for sequencing, you're not going to have any left to use after sequencing. Generally, you can sequence something once, a culture that you get from somebody or a plasmid that you get from somebody, you can sequence it once, and then be fairly confident that when you grow it up and repurify it, it's going to be the same thing. So you're not going to need to resequence plasmids every single time that you grow them up or anything like that. Now, the company that I use for sequencing is called Genewiz. Let's pull up Genewiz. Now, Genewiz, if you're not in the US, you're going to have to find a company that is local to you. Generally, companies are pretty open about DNA sequencing because, like, you can't really do anything bad with DNA sequencing, so most people don't really care. Um, but, find a company that will let you submit samples and send those samples in, get it sequenced. So when you place an order on a place like Genewiz, sign up for an account. If it asks you stuff like PI or institution, you can make one up. Use yourself as a PI. So PI in science terminology means primary investigator. So whoever is just like the main scientist on the thing, that's the primary investigator. That can just be you. If they complain, you can always use my name. We have a pretty healthy account with Genewiz, and I'm sure they would be willing to honor that. Now, one thing to remember is that Genewiz is a science company. So what that means is that they don't charge you immediately. They charge you after your order is done, and then they give you a net 30. So net 30 in science and business terms means they 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 only ask to pay your invoice 30 days after your order was placed or your order was started. So if you receive a random charge from Genewiz, 30 days later, understand that it's a net 30 payment. They never charged you when you placed your order, not like normal people. So the thing that we want to do is Sanger sequencing, like we talked about. Now they have a bunch of different stuff for Sanger sequencing. And generally what you're going to do is plasmid for this class because we have plasmids. You should have 3 plasmids. You don't have to submit them all for sequencing. You can just submit one. It costs like $6 or $7 or something like that. So yeah, it's not too bad, but if you don't want to submit a lot, that's totally okay. So DNA type, just keep to plasmid. What the hell is that reward thing getting in my way? Get out of here, rewards. DNA type, you could just keep the plasmid right here. Service type, um, what we're going to use is premix. Number of samples, put your number of samples. How many are you going to submit? Let's just say we submit one. All right. So let's say we're going to submit P Dawn. So you put in there P Dawn. Now, the length of our, I never know if this is length of the DNA you're submitting or length of the sequencing product that you want. I don't think it really matters. I just usually put in 500 to 1,000. Now, primer. If you're having something custom sequenced, you might have a custom primer. So you will include your primer with the sequencing order. In this case, we aren't going to send any primers. We're going to use some of the primers that are already that Genewiz already has. And so this place where it says my primer name, just leave it blank. All right, just leave that blank. Don't worry about it. Now, when we choose primer selection, this is where you can choose a bunch of different primers that you want. A bunch of different primers that they have in stock that they'll use for you free of charge. Now, if you downloaded SnapGene, what we can do is we can look at the plasmid and you could see which of these primers will work for us, right? So let's open recent. Let's open P-Dusk. Even though we said P-Dawn, they're pretty similar. All right, so let's look at the plasmid map. Let's see what we got. All right. So let's open it up. All right. So the first thing that we have right here that we can see is the T7 terminator. If we go back and look at the primers, we know they have a T7 terminator, so we can always choose that, right? T7 terminator, what it'll do is it should do a reverse sequence through the gene, and in this case, in this P-Dusk plasmid from AdGene, it doesn't have the gene that should be inserted right here, P-Dusk. I mean, DS Red Express 2 or some bullshit. Rita, if you're listening, it's not really bullshit. But we scrolled through, we got F1 origin, we got kanamycin, we got BOM and ROP, which are for plasmid replication. We got the LAC promoter. We got the T7 tag leader thrombin site. If we look at primers, we don't really have any of the rest of these in them. So mostly if you're working with a bacterial plasmid, the ones that you're generally going to see are M13 forward and reverse, PBAD forward and reverse, T3, T7, and T7 term, right? In this case, it's T7 term. They will add that plasmid for us. You don't have to worry about anything else, right? Now, it should be T7 term for all the plasmids we have in the class. So just use the T7 term when you send it off for sequencing. Click Save and Review, right? And the total cost for this is $6, right? So what you have to do then is you have to ship your order to Genewiz. So you add it to your cart. It gives you all these options. Choose to pay by credit card. Right here. Stop freaking out. Choose to pay by credit card. Choose your credit card and pay. It should give you an address here. It's the same address that's in the file on the Google Drive. And that address you should ship your sample to. Now, generally, I would encourage you to use USPS 2 to 3 day shipping, which is, I think it costs like $6.50 or $7. But your samples get there faster, you get your results faster, and the chances of your shipment being lost or, lots of crazy shit happens when you ship first class. If you're pinching pennies, first class is only like $2.50 or $3. So you'll save, $3, $3 to $4 if you ship first class. But generally, I'll ship it USPS, small flat rate, 2 to 3 day priority, which is, like $6.50, $7 or something like that. Yeah. Once Genewiz receives your sample, generally you get the results back within a day, which is really nice. They get you your results back fast, so then you don't have to wait around too long for it. It's a good service. Now, Genewiz is not the only company in the U.S. that does DNA sequencing. In fact, there are a ton. There are a ton of companies, and I don't want you to feel like you have to use Genewiz or Genewiz. Yeah, if you get asked to provide a name of your institution or your PI's full name, like I said in the beginning, you can just make up your institution name. It doesn't matter. You can be the PI. So the PI is the primary investigator. So you can just put your name. If you can't use your name or they don't accept your name, you can always use my name, Josiah Zahner. Like I said, I have a pretty, long-standing account with Genewiz, and they're pretty open, so they probably won't care much. But, first I just try it using your name, and, if you're going to be doing this stuff anyway, you should probably have a fake company because every company asks you to have a company for this stuff. So just, come up with a company name. What's your future biotech company going to be? And use that name. Some companies are pretty hardcore about it, but most companies don't really care. You generally can tell pretty quick which companies are hardcore. But, it's not illegal to lie. It's not illegal to tell them, your institution is biohack watermelons or something. Like, they don't care. Most of them, it's just like a leftover from the age of that academics and, companies were the only people using these services. But now a lot of people are using these services. So companies are starting to become more and more open. But I would still recommend, I've definitely had companies reject me, and I run a fairly decent business, have a bunch of employees, and still companies won't send to me. They're just very skeptical. They think I'm going to hurt myself with like water or something. It's pretty ridiculous. So it doesn't hurt to like buy a domain name, set up a website, set up an email address that you can use that's not a Gmail address. All in all, it probably costs like $20 to do that. And it makes you look a lot more legit, so you can order from a lot more different places. But generally, for stuff like DNA sequencing, you're always just going to be able to find another company. So if somebody rejects you, just search DNA sequencing on the internet, and there'll be another company that'll gladly take your order. Doesn't make any sense why somebody would reject somebody from using DNA sequencing, as like, what are you going to possibly do bad with DNA sequencing? It doesn't make much sense. But, the biotech world, people freak out a lot. So the biggest companies are most problematic to deal with. So like Sigma, um, Fisher Biosciences, VWR, places like that, they're generally a lot harder to deal with than smaller companies. Because generally smaller companies, they don't care. They're just trying to make money. There is no government regulation they're concerned about. That's the thing, is that like it's not illegal to order DNA sequencing. It's not illegal to order DNA. It's not illegal to do anything that we do in these classes. But like, you have to understand that with new technology and when people start getting into new stuff, like people freak out. They would totally freak out, right? That's why like the US government and governments from other countries have come after me multiple times. Even though I do absolutely nothing illegal, absolutely nothing. I make sure I don't do anything illegal, yet still the governments come after me, right? So it wouldn't be crazy if they come after you, right? Not for anything you did wrong, just because people are freaking out. But, in 5 years, this is going to be the cutting edge. You're going to be so far ahead of everybody else because you know how to do this stuff, right? You're going to be able to get jobs doing this stuff if you don't already have one, because this stuff is the cutting edge stuff, right? Everybody taking these classes are the first people in, the general population to learn about this stuff. So it's like you're at the cutting edge. So smaller companies, Genewiz, I recommend. They're fairly smaller. IDT synthesis, and I don't know if they do DNA sequencing. They do DNA synthesis. They suck. Don't order from them. They won't send you DNA. Elim Bio, Elim Bio, they're pretty good. They'll do DNA synthesis if you want DNA. Genewiz does DNA synthesis also, but it's mostly just small primers and stuff like that. So, a lot of it's just trial and error. You know, some some companies that I try to purchase from, other people don't have problems with. Some companies I purchase from, other people have problems with, right? So it just really, really depends. Uh, yeah, it really depends. So it's hard to tell. But yeah, GeneWiz is generally pretty good. They generally don't really care. Um, for chemicals like antibiotics and stuff, Gold Bio, GoldBio, Gold Bio. I'm pretty sure will send almost anybody, and they have like the best prices in the industry anyway, besides the Odin. So, get stuff from there. But yeah, generally it's just, trial and error, finding out um, who will send you stuff and who won't. All right. So when you send your samples in, um, generally you want to send about 10 to 20 microliters of your plasmid DNA. It's better to send more in case they have to So what they'll do is if the sequencing doesn't work at all, for free, they'll try to sequence it again in case it's an error on their end. So if you send more, there's a higher likelihood chance that they'll be able to sequence it again, and a better chance they'll be able to get results. But don't send like so much that you send all of your all of your plasmid, right? So don't go sending like 30 or 40 microliters of your plasmid. That's not really needed. They'll never use that much. Uh, Generally, 10 to 20 is the max you're ever going to need to send them. So, you know, keep it down around there. And it depends on how much plasmid you have. If you have a lot of plasmid, if you have 50 microliters, feel free to send 20. If you have 30, it doesn't matter really if you send 10. So, be stingy with that plasmid because you're going to use it for other stuff in the class, and it does take time and effort to purify plasmid DNA, right? If you haven't purified it already, but it's going to take it's going to take a little bit of time and effort. So, value your time. Yeah. All right. So now, once you submit your samples, what Genewiz is going to do is they are going to send you back a couple files. So in the Google Classroom, there should be a folder that you can go to that's called sequencing, and it should have a couple different files. And those files, the two files that they'll send you are a .seq file, a .seq file, a .seq file, and a .ab1 file. Now, the .seq file can be opened just by WordPad or Notepad, or Snapgene if you want. But what this is this is the DNA sequence that they actually read from your sample. So this is the A's, T's, C's, and G's. So the way Sanger sequencing works is they build a DNA sequence that is they build a DNA sequence by adding successive base pairs. Wait, Sanger. So, hold on. First, let's just go over this. So, this is our DNA sequencing file that they send us. Now, I told you it's just going to be A's, T's, C's, and G's, but I lied to you a little bit. So there's also going to be one other letter in there, and that is N. Now, what N means is it just means that they couldn't determine what the DNA base was for that sequence. Now, what you're going to do is you're always going to have a large number of N's at the beginning and at the end of the sequence. This is just because the error rates get higher at the beginning and at the end. That's all. The ability to call what nucleotide it is gets difficult at the beginning and at the end. Also remember that it's not going to sequence your primer. Your sequencing primer is what binds to the DNA, so you're not going to receive that sequence back. So you're going to receive everything after the sequencing primer. Now, the way that Sanger sequencing works is that you add this primer, and then you add a DNA polymerase, and the DNA polymerase is going to add nucleotides, right? It's going to do like a normal replication. It's going to replicate a complementary strand of DNA that's complementary to the piece of DNA that you submit, right? So when you are replicating DNA, what happens is each base is added on, right? So each new base is added onto the piece of DNA, and the way that Sanger sequencing works is you have bases that have a readout so that when a base is added, we can tell which base is added by fluorescence. So what happens is we get an actual readout of the DNA sequence. Now, this is what is in the AB1 file. They call this a chromatogram, a sequence trace, an electropherogram, I think. All right. Now, each color on here represents a different DNA base, right? So each base has a specific label, like fluorescent label on it. Let's say A. I don't know if these actually correspond to the colors or if they're just, the colors, but A is represented by green, G is black. No, there's no black fluorescence, so T is red, C is blue. And see, you could see here at the beginning, right? Everything's a mess. We're detecting all different types of bases here, right? We can't tell what's actually going on. But you can actually see, look right here. It looks like we have a pretty big green peak, right? Same with here, a pretty big green peak. So green is associated with A. So if you were to say that these two bases are actually A and not N, your guess would probably be pretty good, right? It probably wouldn't be too far off. And we can check and we can compare it to the sequence, which we will do in a second, right? But this is a way, if say you have an N in your sequence that you're trying to figure out, you can actually go into the electropherogram and decode it. So say in our sequence right here, right? We want to see what this, and even though we're getting a signal also from T that's bleeding over, we can probably say that this is probably A. And this N, we could probably say that this is G, and there's a good chance that we are correct. Now this one's a little more complicated. If you look at this N right here, right? This N, it's really hard to tell. There's not a clear signal what it actually is. So that one, we can't just tell by this, chromatogram, electropherogram, whatever you want to call it, by this thing that the sequencing company tells you. Now, the reason these N's are in here is because they usually use automated software to tell these things, right? And automated software has certain parameters that it's going to use to decide what is an A, T, C, or G, or an N. So even if you have an N, or even if you have, say, the wrong base in the DNA sequence, it doesn't necessarily mean that you actually have the wrong base. You can come back, look at this chromatogram, and see if the wrong base is actually there, right? And figure it out. Because you might be surprised that sometimes the parameters on these computer programs are set too high, and they'll only call bases when they're like something like this, right? Like, look at that G. There's probably no question whether that's a G or not. But when you get to other stuff, sometimes it gets a little more questionable, like around here and here and stuff like that. So it's not perfect. None of these experiments are perfect, so they're open to interpretation. If you're really questioning some of the results that you receive back, you can ask Genewiz and generally other sequencing companies to resequence it, and generally they'll do it for free. So what do you do when you have your DNA sequence and you want to see if it's the correct sequence? You want to see if you sequence the correct thing. Well, the first thing you can do is use BLAST. So BLAST. So if you search on the internet, BLAST, NCBI, the basic local alignment and search tool. Now, what this does is it tries to match your sequence against all known sequences in the government database. Generally, you want to try to leave out as many of the ends as you can. So start after all the ends and try to end before all the ends. Copy this, and we'll paste it in. So this will take a second. It's searching and searching and searching. I actually wonder if this will come up with anything. All right, and here we go. So now what happens is we get matches to all the sequences in the database. Now, there's a couple things that you have to know here. A couple things that are important. So when you send something through BLAST, there's two general numbers you care about. One is query cover, and that's the query coverage. So that means like how much of the sample you submitted is being matched to the thing in the database. Because sometimes you submit 100 bases and only 80 of those match something in the database. So sometimes your query coverage is small. Sometimes it's only like 80 or 70 or 60%. But you want it to be as high as possible. You want it to be 100% because that lets you know that the thing that you're matching, there's something that exists that is almost identical. The other is the percent identity. Now, the percent identity is how close is this DNA sequence to the DNA sequence I'm matching it against, right? And identity also takes into account like the similarity of the DNA bases, right? So it's like A is closer matched to T than G is to T. So if your DNA sequence has a T where an A should be, it's going to be more. Oh no, that's similarity. Never mind. I'm thinking of similarity, not identity. Percent identity is just straight match. Do the DNA bases match, not percent similarity. Sorry, it's hot in here. It's getting to my brain. Oh. Any questions been going on? All right. So the role of the primer, sorry I missed that question. When you're sequencing, the role of the primer is to allow the DNA to be amplified. So whenever DNA is replicated or amplified, it needs a primer. This is just the requirements of the machinery. You can't amplify a sequence without, well, you can technically. You can use random primers. So they do have random primers that you can use to try and amplify random sequences. But that's not something that you'd really want to do in this case. It doesn't work out well. You want all the sequences to start at the same place so you can figure out, the structure of the sequence. So you need a definitive primer on what this is. Anyway, so what we have here is we match to a synthetic construct and a cloning vector. So basically a cloning vector is what we're using in these classes. That's a plasmid, it's the same thing. Cloning vectors are things that you put genes into, right? So what this class is about understanding if you wanted to take a DNA sequence, you wanted to take a um, a lizard and turn it into a Tyrannosaurus Rex or something, and you wanted to insert a DNA sequence in there using a plasmid into the lizard, this is how you do it. First, you take that DNA sequence, you put it into a plasmid, and then you take that plasmid and you put it into the organism. In this case, the plasmid's already made. We're going to make our own plasmids later and learn how to make custom plasmids. But we're using plasmids already made, and we're using bacteria as our organism to modify. So cloning vector is the same thing as a plasmid. When we get matches for cloning vectors here, it's the same thing as the plasmids that we're using, so don't worry too much about it. And generally, you're probably going to get a cloning vector match back when you sequence a piece of DNA, because your DNA is in a cloning vector. But generally, you'll also get back a DNA sequence if you're looking at your gene of interest. In this is a sequencing of some random vector that we have that we use as example data. So this is example data. We can see down here that where our sequence is mismatching is where the ends are. N. When you have a dash like this, what that means is that there's a base missing. So there's a DNA base missing here. We can actually go look at that in our chromatogram. So let's copy the sequence. Let's go to our chromatogram and let's find out where this is. So this N, it looks like it's difficult to tell. Off the top of your head, you might say it's blue and a C, but that would be wrong. We're seeing right here it's an A. But what might have happened here is you can see how this hump is really big, and you see how this goes AA right here. So a lot of times when you have extended humps like that, right, like here, TTT, AAA, you can almost see right here how there's two peaks. So one of these peaks might have been missed, and this blue is actually C, right? So we have AAC right here. So our sequence might probably still in fact be AAC. What you can do is you can ask the DNA sequencing company to resequence, right? And see if that clears this up. But you know, it looks pretty good like that, right? That's a pretty big peak right here. It's, you know, almost the same size as this one, if not bigger. And the height is pretty similar. Yeah. So, and we can also look at this one. Let's look at this one right here. This N. So we copy and paste the sequence right before it. NGG. So what we have an AGG. So this one definitely looks like an A. This N, you probably could say that's an A. This one looks like a G. We're supposed to have a double G there, and it's hard to tell. There might be two peaks there. There might not be. You can look at this double G over here, how that peak is, and it looks similar to this peak right here, right? So maybe the double G is also there. The program is just reading this chromatogram wrong, right? You see, makes sense. So this is how you compare sequences. Now, if you already have the sequence, let's say it's a custom sequence, and the sequence might not be in the BLAST database, what you can do is you can select align two or more sequences using BLAST. Now, what this does is it allows you to paste your sequences. So let's take our query sequence, and then in the Google Drive, there should be this DNA, open human DNA. This is the actual sequence that we're trying against, right? And you can just press Control A, Control C, copy this whole thing, paste it in the second box, and then run the blast. Now this will try to just align these two sequences to each other and see if they match in any places. Any places on the sequences match, right? And how well they match. Yeah. So it says we're only getting 70% query coverage, 98%, 99.87%. So how big is our sequence? It seems weird. It should match. Anyway, so we look through our sequence, and do we have any mismatches? It probably cut off the mismatches at the end. What we can do is we can go back, let's edit our search, and instead of doing highly similar, let's say more dissimilar sequences. It's optimized for more dissimilar sequences. Yeah, so this, well, let's see. It still only says 77%. Why is it only doing 77%? Why is it matching the whole thing? What's going on here? Ugh. Edit search. How big is this? Let's open it up and let's just open this up. How big is the sequence we're trying to use? Is it only 700? I thought it was more than that, but who knows? Sequence. So let's start after the ends we started, and we went probably to somewhere. Yeah, it should be like 1,000 bases. I don't know why it's not matching all this stuff. All right. Let's try this one more time. Edit, copy. Let's submit. Let's try somewhat similar sequences. All right, we're going for it. We're going for somewhat similar sequences. Only somewhat similar. No, it still only gave us 70% coverage. Yeah, it's acting weird. I don't know what's going on. Anyway, we could see that this matches our plasmid between 1 and 785. So let's see. So 1, we started right here, and 785 in our sequence is approximately right here. Now let's take that. What we can do is we can go into Snapgene and we can just search. No matches were found. God damn it. Is there like a random N in here somewhere that I'm never going to be able to find? Probably. All right, let's just try just the end. Let's see where it ends. All right. So it is ending right here. After we see TCGGG, let's see if that matches in our sequence. Yes, it does. but it seems to be going in the opposite direction. Is this the reverse? Anyway, you get the picture. You understand. This is generally how you match up your sequences. You figure out if the sequence you have matches your plasmid, if you have the correct thing, if you inserted the correct genes into your plasmid, right? And so we match that up. Now, one quick thing that I just want to show you is what a bad sequence looks like. So we also have this, bad DNA sequencing data. Yep, it's real data. It's my fault. Actually, I don't actually know what happened with this. Now, you might actually get back sequencing data from a bad sequence. And what happens is, actually, this isn't that bad. There's some ends in it and stuff like that. But generally, a DNA sequence is really good. Now, you can see here, remember, like we learned, you could call this N probably a T. You could call this, you might not be able to call that N. These N's, maybe a C, maybe an A. But generally, if you're not sure, if it doesn't look definitive, just ask them to resequence it or something like that. All right. So this week, if you haven't purified your plasmids, what you should do is you should purify those plasmids, and then you should send them off for DNA sequencing. It is only $6 plus shipping, which is really inexpensive, right? To sequence your own custom DNA or the DNA of these plasmids. And it helps you develop a relationship with these companies. And, it gets you moving towards doing all your own genetic engineering at home or wherever you are. No, so what they do in Sanger sequencing AAV, you ask when they add the next base, I'm assuming they remove the fluorescence from the previous base. Why then do we not get that buildup? No, what happens is each base, I failed to mention, it has a terminator on it. So it can only add one, they add terminators so that what happens is the sequences get stopped after a certain amount of time, right? So if we have a bunch of things amplifying and we put in an A terminator, we put in a G terminator, we put in a C terminator, what happens is they're all going to be stopped at random points in their sequencing, in their amplification replication process, right? And what happens then is we have a bunch of sequences that we know how long they are and what the fluorescence is at that point. And so we then can tell what the base is at that point, right? And you get a whole lot of signal. That's why you get a bunch of, different things, because sometimes you'll get wrong bases incorporated, wrong other things and stuff like that. So yeah, so it's like you just if you really want to know more about Sanger sequencing, obviously just look up the Wikipedia page. It uses this dideoxy terminator method, and I'm sure it'll explain it way better than I can. But yeah, it'll help you gain insight into why maybe you're getting those errors in the background signal and all that other stuff. Any other questions? Any other questions? Yeah, So make sure you submit your DNA this week. It's really cool. Let us know the results. Let us know if it worked out. I want to hear if it worked out. I want to hear what's going on. Let me know if you have any trouble submitting. If they hassle you or anything, just feel free to let me know. Yeah, but let everybody know your results. Let people know how your experiments are going. Cool. Glad you ordered a mini prep kit. But yeah, you know, get some results and share them with the class so other people can also learn from you. Anyways, appreciate you all coming out this week. If you have any more questions, ask them in the Google Classroom. Get those experiments going. And I will talk to you next week. They sent you a plant genomic extraction kit. That's unfortunate. I'm sorry, SnapVault. Got a plant genomic extraction kit instead of the mini prep kit. We'll try. Another great class. No problem, Kent. All right. Alexis, I don't really know what your question is asking. Robotic automation will have no effect on biology. People who think it will are just ridiculous. They've never done experimental biology. What are some areas or applications in which we can get work on? I don't. You mean work in? All right. Well, I'll see you all next week. Thanks for coming out. Asking questions in Google Classroom. It helps everybody. Laters.